or digestion occurs by the process of hydrolysis. And hydrolysis means a couple of things here when we're looking at digestion. Water itself is split, but in the process, the water is breaking the carbon-carbon bonds between sugar groups, or between glycerol and fatty acids, or between amino acids of a protein. So if we look at that process, here we have a carbon-carbon bond, and to that we add water. This is a reversible reaction. So what we get then is the carbon is split, and we add the hydrogen, one hydrogen, and the hydroxyl group to the other. So the interaction between water, the polar nature of the water, the charges on hydrogen and oxygen interfere with the sharing of the electrons between the two carbons and interrupts that bond created by the sharing of those electrons. So water is split, yes, but it also breaks, slices, if you will, the carbon-carbon bonds. All right? So this is a rather slow process. About, I don't know, 16 or 17 years ago, when I first started teaching here, I was able to check out a video, and I looked for it online last night, and I couldn't find it or couldn't come up with the proper search words. But a piece of meat had been sterilized, placed in a mason jar of sterile water. The jar had also been sterilized, and then time-lapse photography over a period of about a week and a half, 10 days. And on the surface of that meat, the water was interacting with it, and you could see the water start to grow cloudy as the proteins were being broken down into smaller peptides. But it took forever. And of course, the meat was the large surface area. It wasn't ground down just to be visible in the bottle. But it still would take a long time if we waited for it to occur with just the water and the food substrate. So our body produces enzymes. And as you know, enzymes work on the substrate. The substrate would be the carbon-carbon in our water, and increases the rate of the reaction. But it doesn't become a part of the product. Okay? It just brings the components of the reaction closer and strains the bonds so that they're more likely to interact with the other molecules. And so we're going to look at, first of all, the basic chemistry of how starches are broken down to simple sugars. It's a two-step process, and the same is true for proteins. That's a two-step process. And then we'll look at what happens to triglycerides. When we're done with that, we'll then take that process and see what happens in the mouth, what happens in the stomach and the small intestine, and the hormones that are involved in controlling that. So we're starting out with carbohydrates. And carbohydrates are polysaccharides. So we have two steps. An intermediate step that uses the enzyme amylase, and then a final step that occurs with the final enzymes that are associated with them. So let me, this is kind of a simplistic view. Not all of our simple sugars are six-sided. Fructose is a five-sided ring, but for simplicity, I'm going to draw circles on the board rather than take the time to draw the six sides. So we start out, notice it's a two-step. We have intermediate digestion and then the final digestion. All right? With intermediate digestion, our products, the disaccharides, are of too large a size to cross the plasma membrane of the cells. So they're not absorbed. And as I indicated earlier, you can chew crackers and get a sweet taste, but you're not going to transport those disaccharides into your mouth, across the epithelia of your mouth. So we start out with polysaccharides, which is what starches are. So this could be cornstarch or flour or some complex polysaccharide. So a series of sugars. So each circle represents a monosaccharide plus water. I usually forget to put that in there. And then we add our enzyme. So in this intermediate step, the enzyme is amylase. Amylase has two sources. We talked about salivary amylase being primarily secreted by the parotid glands. And our major source is, as we'll see a little bit later on, is from the pancreas. Okay. So then our product from this is going to be disaccharides and oligosaccharides, which are anywhere from about 8 to 10, rather than 20 to more than that. And we're going to just follow through with the disaccharides. If you've had nutrition, this should be a simple review. This could be 30 or 40 off here. So that's the intermediate step. All right. So it doesn't give us an absorbable product. Then we take the product of the intermediate step, we take the disaccharides, and we have three different kinds. So we have sucrose, and maltose, and lactose. Add water. And then each of these has its own unique enzyme. Many times, most of the time probably, enzymes end in ASE. So we have sucrase, maltase, and lactase, being the enzymes. And these are produced by the small intestine. And then our products are going to be monosaccharides. So every disaccharide here has glucose. And then sucrose also has fructose. Maltose has another glucose. And lactose has galactose. So these are now monosaccharides, or simple sugars. And using secondary transport and facilitated diffusion, we can move these out of the lumen of the small intestine and absorb them into the body wall. So this would be the final stage or step of digestion, something that gives us a product that the body can absorb. All right, this is where most of our energy comes from, by the way. It's most of the, unless you're on a high protein diet, most of our intake is from carbs. We have proteins. So a very similar diagram, all right? We have an intermediate step that occurs in the stomach to a small degree, but mostly in the lumen of the small intestine. And then the final step occurs at the microvilli of the small intestine. So again, it's a two-step process. It looks exactly the same as far as my diagrams, which is changing the wording. So instead of a polysaccharide, we have polypeptides. And depending on where our organ is that we're utilizing for the source of the enzyme and in our intermediate stage, The stomach is going to release pepsin. And the pancreas releases trypsin and chymotrypsin. Now notice here our enzymes end in IN. So on the quizzes or on the lecture exam and on your sample exam, there will be a table, and I will fill in part of the table for you, various parts, but there will be the name of the chemical, lipase or gastrin or whatever, and then a column for is it an enzyme or a hormone, and then a column for what organ makes it, what organ does it work in, and or maybe what is its substrate. I think you have a table like that on your lab write-up for today. So if we're talking about salivary amylase, that's an enzyme. What organ makes it?
Salivary gland.
Salivary gland, primarily the parotid gland. Where does it work?
Mouth.
In the mouth. Okay, the substrate would be the polysaccharides. So in the case of pepsin, the stomach makes it and it works in the stomach. And it's an enzyme. Trypsin, the pancreas makes it, it works in the small intestine. Okay, so depending on the, and I would give you, I might give you whether it's an enzyme or hormone, or I might give you the organ where it works. So depending on the molecule, various components. All right, so the result of the polypeptides are peptides, oligopeptides, and dipeptides. So a variety of different amino acids that are still attached to each other. So each of these circles is representing an amino acid rather than a sugar. Okay. So then we bring our peptides over here. So that is our intermediate step for proteins. And again, we're not capable of absorbing them into the body. And so now within the small intestine, we have a couple of, well actually 3 different types of enzymes. We have carboxypeptidases, and we'll look at this in detail when we talk about the small intestine. We have aminopeptidases, and these just work on different ends of amino acids. So we have an amine group, and then we have an acid end, the COOH, our carboxyl end, right? And then we have dipeptidases. And then again, our final product is going to be individual amino acids. Do you remember how we transported amino acids across the wall of the proximal convoluted tubule? We had, for glucose, we used secondary sodium, secondary active transport with sodium. And we use that same mechanism with our amino acids. That's how we move amino acids out of the lumen of the small intestine as well. Okay. So again, just looking at an overall pattern or scheme, and then we'll look at this again when we get to the small intestine. So intermediate step, in the lumen of the stomach, so primarily in the lumen of the small intestine. Final step, right at the microvilli. These enzymes are present in the membrane of the microvilli of the cells covering our villus. Okay, when we look at lipids, there's a great variety of lipids. There's phospholipids and cholesterol. We're going to focus just on triglycerides. We actually have lipase produced by the mouth and the stomach and the pancreas. But again, within the pancreas is the most important source. So again, as I said, we're going to focus on triglycerides. So triglycerides are a molecule of glycerol with 3 fatty acids. We've alluded to this when we talked about ketoacidosis. And I said, as lipids are broken down for fuel, if the body's unable to use the glucose, then the fatty acids are released, and that contributes to ketoacidosis in the diabetic individual. So we have our glycerol molecule. So this is the glycerol. And then to each carbon, I'm not obviously attaching the hydrogen. There'd be hydrogen atoms attached to this as well. We have the fatty acid. So an acid ends in that fashion. All right, so that's what I'm drawing here. So that's the acid end, and then we just have a long chain of carbons after that. And another fatty acid here. Again, there are hydrogens on here. So that's our triglyceride. So tri referring to the 3 fatty acids attached to each of the 3 carbons. So when we take this and we add lipase, then we often get a monoglycerol, which is glycerol with 1 fatty acid. These would be hydrogens. And then a whole bunch of fatty acids. This is the reaction that we're going to be doing in lab today. So I bought a quart of whipping cream last night and brought it with me to lab. And this morning the lab techs added ground litmus powder to it, which is a pH indicator. And so pancreatic lipase, which is our enzyme that we're using, is going to work in an environment of what pH? Acidic or alkaline? Alkaline, because remember the pancreas also releases bicarbonate ions. All right? So hold on just a second. I'll show you what that looks like. So this is whipping cream to which powdered litmus has been added. And you can see this light lavender color indicates an alkaline pH. All right. When if digestion occurs and fatty acids are released, we would expect the pH to drop and our pH indicator to move more to the reddish to pinkish mauve, if you will, color. So that's how you're going to determine whether or not digestion has occurred. We have 3 different incubators. The ice is over there. You would expect at 0 degrees that there's not much molecular movement. And so it wasn't until the very end of checking the times that one of the test tubes, in fact the student didn't even notice, she was carrying it back to her desk. I said, you've got some digestion there. She said, no, none of these are. I said, hold those 4 tubes up together, and one of them was just starting to turn pink at the very end of her experiment. Okay? And that's due to the release of the fatty acids. Let me put this back in the fridge and then I'll be right back. Okay, so now let's go through our digestive organs and talk about which of these 3 different foodstuffs have any type of intermediate or final step digestion occurring. All right, so in the mouth, our pH is almost neutral, okay, just slightly acidic from the saliva. Our mouth makes 2 enzymes, salivary amylase and salivary lipase.
Professor,
Yes.
on the notes you put 6 to 8, so it's 6.8.
Yeah, there's a range, but the average is about 6.8.
Oh.
Salivary amylase is going to work on carbohydrates. So we do have some intermediate digestion for carbohydrates. But only for as long as you swallow. All right?